Afternoon, Doc.
So we have new student from this class. Alexander. Supposedly, there should be 18 students in this class. Michaela, are you in? Alabado?
Yes, Doc.
Nor Osmati Barhama?
Yes, Doc.
No. Gabriela Karag?
Yes, po,
Grace?
Doc. Yes, po.
Pian Pian? Chow?
Yes, Doctor.
Pian Pian?
Yeah.
Okay. Alexander?
Yes, Doc. Good afternoon po.
Abdul? Pax, are you in? Luansi? Lixwansi Duan? Lixwansi? Duan, are you in? Lixwansi? Earl Kirsten Roth Gamboa? Are you in, Earl Kirsten? Kenneth Dido? Are you in? Wang Ning? Are you in, Wang Ning? Sura?
Yes, Doc.
Miyuan Lu? Are you in, Miyuan? Gemela,
Present po.
Ever, Reyes,
Present,
Joyce,
Doc.
Peter Samonte,
Present, Doc.
Maria Salome. Maria Salome is still out. Jem Taladay
Present, Doc.
and Sing Yu. Sing Yu. Okay. For those who are new student in this class, welcome. I welcome you. Nur Osma. Nur Osma. Nor.
Hello po. Hello, Doc.
Okay. Where do you work again?
Ciudad Medical Zamboanga po.
Okay. Pian Pla. Pian Pian. Chow.
Yes.
Pian Pian, where do you work?
Yeah. I'm here.
Yes, Pian Pian. Where do you
Yeah.
work? Are you working in a hospital?
No, I'm no working.
You are not working. Okay.
No. Yeah.
How about you, Liswan C? Liswan C. Are you in? Ning Wang. Ning Wang.
Yes, doctor.
Where do you work, Ning? Ning Wang, where do you work? In China, in mainland? Are you working in a hospital or a clinic?
Yes, sir.
Miyuan Lu? Mayuan, are you in? Mayuan Lu? Miyuan? And Sing Yu. Sing Yu.
Yes.
Okay, Sing. Where do you work, Sing? In China?
Yes.
Are you working in hospital or in a clinic? Sing Yu.
And yes.
Hospital.
Yes.
Okay. I have already. I want you to go to the Google Classroom, and then you have to access the syllabus. And I hope that some of you have already the syllabus. By the way, have you accessed the Google Classroom and have you seen the syllabus? New student, you have
Sir.
to. Yes, Jamila.
Sir, I don't have access po in molecular on the Google Classroom.
Why? How come? You do not know the code? Why, Jamila? You have not yet, Jamila, you have not yet seen the You have not yet seen the syllabus?
I have po, but the Google Classroom, I have trouble going in.
Why? What's the problem? The code? Do you have the code? None?
I
Jen?
have, sir, but I will try to go in today.
Okay. Okay. The code is there, sent by Earl. The code so that you could enter the Google Classroom. Vincent? Are you in, Vincent? Vincent, are you in?
Yes, doctor.
Okay, Vincent. Do you know some of the students from mainland China? Vincent,
Yes, I know them.
you know them. Sora, you know
Yes.
them. Okay? All of you, most of you is working on the same hospital or what? Are you working in the same hospital or different hospital, Sora?
Actually,
different hospital.
A different hospital. Okay. Our class is about biotechnology. Last time, the subject that I told is about advanced biochemistry. This is the first time that I told this subject, application, biotech, principles of biotechnology and application and its application. Okay. If you have access the Google Classroom, I have already uploaded the PowerPoint presentation, the introduction, the PowerPoint presentation, which is the introduction to this course. And I am requiring all of you to have a copy of that PowerPoint presentation of mine. Okay. Most of you here have taken advanced biochemistry. And there are some students, nor Osma, nor have you taken, you were not enrolled in my advanced biochemistry class last time, nor?
Yes, doc.
Are you enrolled? No,
Not po, not.
you are not enrolled. But you
Yes.
know PAX. Do you know PAX?
Yes, doc.
Okay. I think PAX was the one who encouraged you to enroll in the graduate
Doc.
school. Okay, good. Try to encourage more nor to enroll in Master of Science in Medical Technology in TUA, Trinity University of Asia. Like now, there are about 5 students from mainland China. From mainland China. And it's good that I welcome you, students from mainland China. I hope that you will learn something from this course, which is also very important. It is very important subject today, biotechnology. But again, I'm going back to my lecture, to my previous lecture in advanced biochemistry. I want to review the topic on nucleic acid, because if you are going to look at the syllabus, nucleic acid is the foundation. Nucleic acid is the foundation of this course. You will be dealing about nucleic acid, its structure, its properties, its characteristics in the succeeding lecture of mine. We will be dealing with genetic manipulation, how we can manipulate the DNA, how we can manipulate the organism. And so in this case, I would like to have a review on the structure of the nucleic acid. The structure of the nucleic acid, how the nucleic acid is synthesized. Nucleic acids are synthesized or formed again. And I hope that those who have attended my class in advanced biochemistry, you could still remember the structure of the nucleic acids and their properties. Okay, this is just a review class, and for those new students, the primary reason why I have to review this again, I have to discuss this again with you, is because we have new students who have, I think, very little or limited information about nucleic acids. So I have to review this again. We know that the nucleic acids, these are the building blocks. The building blocks of the different processes involved in living organism. And we know from the previous lecture that the DNA and RNA, it is very important in the synthesis of protein. Okay, again, just a review for those who have limited knowledge about DNA, for those new students, that the DNA and RNA, one of the components of DNA and RNA is sugar. And this is the deoxyribose sugar and the ribose sugar. This is one of the components. With regards to the DNA, the sugar that is involved is called 2-deoxyribose. Again. And the sugar involved in RNA is known as the ribose. Okay? And you look at the difference again between the deoxyribose and the ribose. The sugar found in the DNA and the sugar found in the RNA. Okay, you look at again the difference. The difference is with regards on the second carbon atom. First, you must understand that this sugar, it has 5 carbon atoms. 1, 2, 3, 4, 5. It has again 5 carbon atoms. That's why this sugar is also called pentose, because it has 5 carbon atoms. And the difference between the two sugar is that in the second carbon atom. If you are going to look at the second carbon atom class, again, you will note that it only contains hydrogen and it does not contain oxygen. That's why it is called 2-deoxyribose, meaning that the carbon to represent the second carbon atom, and the sugar is called deoxyribose because it does not contain oxygen. Because the hydrogen, it does not contain oxygen. It only contains hydrogen. Okay, that's why it is called deoxyribose, from the word de, meaning absence of oxygen. With regards to ribose, as you can see, you look at the second carbon atom, it contains a hydroxyl group. Okay? The second carbon atom contains a hydroxyl group, and since it is hydroxyl, it has oxygen. And that's why it is called ribose. Okay. So there are 5 carbon atoms in the sugar. And the difference between the two sugar is with regards to the presence and absence of oxygen in the second carbon atom. This is how the two sugars differ from each other. So if it is a DNA, the sugar is called deoxyribose. And if it is an RNA again, it is called a ribose. And this is one of the components of nucleic acid. Okay? RNA. So these are the two types of nucleic acid. the DNA and the RNA. Again, I would like to explain again, I would like to explain to you the monomer structure of the nucleic acid. So it has the sugar now. It has the sugar. And since the sugar does not contain oxygen in the second carbon atom, we call that a DNA or deoxyribose. The sugar is a deoxyribose. Again, another component of the nucleic acid is the phosphate group. The phosphate group is attached to the fifth carbon atom of the deoxyribose, of the sugar, the fifth carbon atom of the sugar. And take note, class, that the phosphate group, it is negatively charged. It is negatively charged. And the phosphate group is linked to the 5 carbon atom by means of a bond known as phosphodiester link. Again, phosphodiester link. The attachment of phosphate group to the sugar is brought about by the phosphodiester bond or phosphodiester link. That's why the DNA, it is carrying a negative charge in physiologic pH. Okay, so these are the two components of the nucleic acid, specifically the deoxyribonucleic acid, and this is the nucleoside. The nucleoside, and you call that 5' deoxyribose monophosphate. It is called monophosphate because there is only one phosphate group. There is only one phosphate group, and you call that 5'. It is 5' because the phosphate group is attached to the fifth carbon atom of the deoxyribose. Now the nucleotide structure. We have now the sugar, the deoxyribose. We have the phosphate group, and there is another component of the DNA, which is the base. Okay, the DNA and RNA has with it a base, and this is known as the nitrogenous base, which is attached. This nitrogenous base is attached to the first carbon atom of the pentose monomer, pentose sugar. And there are two types of these bases. One, again, you call that the purine bases. The purines. And one example of purine base is known as adenine. It is called adenine. Again, as you can see, that this purine-based adenine is attached to the pentose sugar by way of the 9th nitrogen atom and first carbon atom. The 9th nitrogen atom and the first carbon atom. Okay. So these are now the components of DNA, the oxyribonucleic acid. And what do you call this one? Take note of the adenine base. The adenine base, it has an amino group. And it has five nitrogen atom. It contains 5 nitrogen atom. And the rest, they are carbon atom. Where can you find the carbon atom? In 2, 4, 5, 6, and 8. Okay. So there are 8 atoms. There are 8 atoms, and 4 of them are carbon atoms. 4 of them are carbon atoms, and 4 and 5, they are nitrogen atoms. There are 9 atoms in the structure of adenine. 9 nitrogen atoms, 9 atoms in the structure of adenine. And there are 5 nitrogen, and there are 4 nitrogen, 5 nitrogen, and there are 4 carbon. There are 4 carbon. There are 5 carbon atoms, and you call this adenine. And what do you call this nucleotide? Adenosine 5 monophosphate. You call that adenosine 5 monophosphate. So again, remember class that the DNA and the RNA, it contains the phosphate group, it contains the pentose sugar, and it contains the base. Okay. And another purine base found in the DNA it's called guanine. Okay. So there are 2 purines, adenine and guanine. The same manner also, guanine is attached to the pentose sugar between the 9th nitrogen atom and the first carbon atom. The 9th nitrogen atom and the first carbon atom. And you call this one, this is the guanosine 5 monophosphate. guanosine 5 monophosphate. And this type of DNA is made up of phosphate group, the pentasugar, the oxyribose, and the base known as guanine. So this is the structure of the DNA. And then the next group of bases, you call that the pyrimidines. The pyrimidines. The structure of pyrimidines is different from the structure of purines. Okay, so this is the structure of cytosine. The cytosine class, it has an amino group. It has an amino group, and it has only 6 atoms. It has 6 atoms. And in the structure itself, it has only 2 nitrogen atoms. It has only 2 nitrogen atoms in the structure itself. 2 nitrogen atoms. And the rest, they are carbon atoms. And you call this type of nucleotide, it is known as cytidine 5 monophosphate. You call that a cytidine 5 monophosphate. And the other one, the other pyrimidine base, it is known as thymine. This is another pyrimidine base known as thymine. The structure of thymine is similar to the structure of cytosine, but it is different with regards to the location of the carbon atom. Okay? Yes, it has also 6 atoms. It has 2 nitrogen atoms, and it has 4 carbon atoms. 4 carbon atoms. And so you call this nucleotide thymidine 5 monophosphate. You call this thymidine 5 monophosphate. And then the last pyrimidine base yes,
Doxygen po. May
Yeah,
kailangan
yeah.
po magpa-accept sa inyo.
may kailangan magpa-access.
Opo, magpa-accept po para po makapasok sa Google Meet.
Well, hindi ba nila hindi nila alam
Sir, ganun din po sa amin. Bago po kami makapag-access ng Google Meet nyo, kailangan nyo po i-accept.
ganon? Hindi lang kayo basta-basta makapasok? Bakit yung iba nakakapasok
Opo.
Yung iba hindi ko na kailangan i-accept pa. Basta nakakapasok lang sila. Miwan. Okay. Okay. Okay, so as you can see in your class, so this thymine now, and then the last pyrimidine base is uracil. And we know that the uracil base So class, the RNA, the nitrogenous base of the RNA, you call that uracil instead of thymine. You call that uracil instead of thymine. And how do you differentiate a thymine from uracil? You call this now the uridine 5 monophosphate. What is the difference between a uracil and thymine? You have to look at the 5 carbon atom. You have to look at the 5 carbon atom so that you could see the difference between thymine and uracil. In the uracil class, the fifth carbon atom does not contain methyl group. It does not contain methyl group, but instead it has only a hydrogen atom. Whereas in thymine, you look at this thymine, they have almost similar. The structure of thymine and cytosine almost similar, except that you look at in the fifth carbon atom. In the fifth carbon atom of thymine, it has methyl group. It has methyl group. And that is the difference between thymine and cytosine and uracil. The absence of methyl group in the fifth carbon atom with regards to uracil. And also, the presence of uracil is an indication that it is an RNA. The presence of uracil and is an indication that it is an RNA. Another indication that it is an RNA besides the presence of uracil is the second carbon atom. If you are going to look at the second carbon atom, it contains a hydroxyl group. It contains a hydroxyl group. The second carbon atom contains a hydroxyl group. That is another indication or recognition of RNA, ribonucleic acid. So there are two bases by which the RNA and the DNA are different from each other. First, the DNA, it only contains hydrogen atom in the second carbon atom of the pentose sugar, while the RNA contain the hydroxyl group in the second carbon atom of the pentose sugar. Another difference is that the DNA does not contain thymine, while the RNA contain uracil. Okay, you have to bear with this in mind again, class. And again, in the structure or in the DNA sequence class, what you can find only is the symbol of the bases. What you could find only is A for adenine, G for guanine, C for cytosine, T for thymine, and U for uracil. And this symbol, this letter, indicates that it is a nucleotide. That if you will see A, which is made up of adenine, meaning that it contains the phosphate group and it also contains the sugar, the pentose sugar. Okay, nucleotide base pairings. Again, I'm going to emphasize to you that this is very important. The base pairings, the base pair. The base pairs is usually adenine and thymine. The pair is always adenine and thymine. This is the complementary base pair of the nucleotide containing the base adenine and thymine. You call that the 80 pair. Another pair is known as the and this adenine, this is the adenine, and this is the thymine. Okay, this is the thymine nucleotide. They are joined together or linked together by means of a bond called hydrogen bond. They have what is known as hydrogen bond. This 80 pair, it has only 2 hydrogen bond. Unfortunately, hydrogen bond is a weaker link than covalent bonds. That's why it could be easily hydrolyzed. And then the other base pair is called the guanine and cytosine, the GC pair. The GC pair. With the case of the GC pair, okay, guanine and cytosine, okay, it has 3 hydrogen bonds. The GC pair has 3 hydrogen bonds. And for this reason, the GC base pair is more stable than the 80 base pair. It has a higher boiling point because of its 3 hydrogen bonds. So it's more stable. The GC pair is more stable than the 80 pair because it has 3 hydrogen bonds. And the RNA base pair, you call that as the AU pair. The AU pair. If it is an RNA, always adenine is paired with uracil. If it is a DNA, adenine is paired with thymine. Always bear this in mind, class. The complementary base pairing. This is very important. The same manner also with the 80 pair. The AU pair, it is linked by means of 2 hydrogen bonds. Okay? As you can see, in the AU pairs, there are 2 hydrogen bonds. This is similar to the 80 pair, which has also 2 hydrogen bonds. Okay. Now you know the pairing with regards to a DNA and the RNA. The DNA and RNA. Okay. And always bear this in mind that if adenine is mispaired to cytosine, there is something wrong. There is something wrong with the complementary base pairing. There is what is known as mispair. The DNA. Okay, so alternating backbone of the deoxyribose and phosphodiester group. Okay, so this is a DNA polynucleotide. It is now a polynucleotide because there are more than one nucleotide. This is a nucleotide. This is another a nucleotide. And in this case, there are 5 nucleotides. And this is a double-stranded DNA. And this is a double-stranded DNA. This is one strand, and this is another strand. And usually, the addition of the nucleotide is from 5' to 3'. 5' to 3'. And this 5 indicates the 5th carbon atom, and the 3' indicates the 3rd carbon atom. They are anti-parallel. Wherein the sugar is outside and the bases, they are inside. Okay, the first one is the AT pair. The second one is the GC pair. The third one is the AT pair again. The fourth one is the CG pair. The GC, the CG pair. The fifth is the AT pair. And the sixth, the fifth is the CG pair. So this is the double-stranded DNA. And you look at their pairing. So the sequence is A, G, A, C, A, C. That is the sequence of the strand. And another sequence of the strand is T, C, T, G, T, G. That is the sequence of the other strand. And this is what you will only see in the literature. In the literature. And this is understood when you see this A and T, that means that it is a nucleotide. And it is pair of adenine and thymine, and it is a nucleotide. When I'm talking of nucleotide class, again, it contains sugar and it contains the phosphate group. It contains the sugar and it contains the phosphate group. Okay? So that is the complementary base pairing. And of course, if you want to denature, if you want to break the bond, the hydrogen bond, what you should do is you just apply heat. Apply heat. And also by using an extreme highly alkaline solution. Highly alkaline solution. That's why in the RT-PCR, the temperature vary during augmentation of the DNA. You vary that there is what is known as denaturation and then renaturation of the DNA. It will separate and then again it will combine based on the temperature. Based on the temperature. At higher temperature, what will happen to the DNA, to the double-stranded DNA? It will be denatured. It will be separate. And then what will happen if you lower the temperature? There will be again annealing. The DNA double strand will be again annealed. That will become a double-stranded DNA. How about the RNA? RNA has a single polynucleotide chain. It is not smooth and linear in structure. Okay, this is the structure of the RNA. Again, it has extensive region of complementary adenine and uracil or GC pairs. That is how the messenger RNA you could see. Can you still recall what is this? This 3-letter word. Can anybody recall that? Michaela, are you in, Michaela? What is this three-letter word?
Codon, Doc.
That is called codon. Okay? That is called codon. And it is very important that this codon, it indicates an amino acid. This is a code for an amino acid. AAC, this is a code for an amino acid also. We call that codon. Codon bases. So what will happen in the RNA molecules? It folds on itself, forming a structure called hairpin loops, random coil, higher order folding. Okay, it is folding, and you call this a hairpin loop. You call that a hairpin loop or random coil. Okay, so that is just a review class about with regards to the structure of RNA and DNA. I want you to look at the literature. I look at the literature in such a way that you will have full understanding about DNA and RNA. Now that I have shown you, I discussed with you about the difference between the RNA and the DNA. Now, if you want to have more knowledge about DNA and RNA, you have to look for literature, books, in any books in biochemistry, in any books in molecular biology, in any books in biotechnology. You could always see this structure, the DNA and RNA. But the important thing, class, is you should know how to differentiate the DNA and RNA. And when is it a DNA and when is it an RNA? That is the most important thing. Okay. Class, do you have any question again about the RNA and the DNA before I will proceed to the next topic of my lecture? Is Vincent around? Vincent?
Yes, I'm here.
Yes, Vincent, the male guy. Sora?
Actually, Vincent is out because of some stuff, so I help his work in this
Okay.
class. Yeah.
But Vincent is not around in our class. Is not present in our class today.
Yes.
He is not present in our class today. Sora.
Actually, Vincent is we do the interpreting work. So
No, what I mean is that he is not attending our class. Vincent is
yes,
not,
yes,
is
yeah.
not
Right now, I help. I assist him about his work. Yeah.
okay. That's why he is not present. Unlike last semester, he is always present in our class. Sora? Okay,
Yes, Doc.
okay. So, how about the others? How about the others? Did you understand? Liswansi, are you present? Pian Pian Chow? Pian Pian, are you present?
Yeah, I'm here.
Yes. Did you understand my lecture, Pian Pian?
Yeah, I understand.
Okay, how about you, Liwansi? Do you have any question? Liwansi. Ningwang? Ningwang, do you have any question?
Yes, Doctor.
Okay. Miyuan? Miyuan Lu? And Sing Yu.
Yes, Doctor.
Okay, if you have any question, you just you just anyway you look at my email. You just email me if you want to clarify. Just send me an email. Student from China, if you want some clarification about my lecture, just send me an email. Okay. Okay, now again, I want to review again about DNA replication. How the DNA is replicated. Before we discuss about DNA transcription again and DNA translation, first we have to review how the DNA is replicated. In other words, how the DNA is synthesized. Okay, the DNA replication is the process of copying a double-stranded DNA molecule. This is the process of copying a double-stranded DNA molecule. As you can see in the diagram, this is a DNA, this is a double-stranded DNA. This is a double-stranded DNA. And this is how this double-stranded DNA is being copied, or what is known as replicated. So in this case, the first thing that happened to the DNA during the process of replication, it has to separate. It has to be separated. And then each strand, this one, each strand will be replicated or it will be synthesized. Or another DNA strand will be synthesized from this strand. And that is what we call replication. In other words, there will be what is known as doubling of the DNA or the strand. Okay. There are several enzyme classes. Again, this is just a review. There are several enzymes that are involved in the process of replication. One primary enzyme is known as DNA polymerase. And these are enzymes that carry out all forms of DNA replication. A DNA polymerase can only extend an existing DNA strand paired with a template strand. It cannot begin the synthesis of a new strand. To begin synthesis of new strand, a short fragment of DNA or RNA called primer must be created and paired with a template strand before DNA polymerase can synthesize new DNA. I discussed with you about this primer. RNA primer. Gabriela, are you in? Gabriela?
Yes po, Doc.
Have you attended my advanced biochem last time?
Hindi po, Doc.
Ay, hindi pa?
Opo.
Are you working Cardinal Santos?
Yes po, Doc.
Okay. Abdul, are you already in? Nachet? Earl Kirstein wrote. Are you in?
Yes, Doc.
Earl, can you still remember what enzyme is responsible for the synthesis of a primer? Rna primer? Can you still remember?
It's the polymerase, Doc.
Is it? The enzyme responsible for the synthesis of a primer? You could not anymore remember? How about you, Kenneth? Kenneth, are you in?
Yes, Doc.
Do you still remember the enzyme that is responsible for the synthesis of
Dna Primase, Doc.
Primase,
okay,
Doc.
primase. You call that primase. Okay. So the DNA polymerase is the one that is responsible for the replication or for the copying of the DNA strand. Whereas the RNA primer is synthesized by an enzyme called primase. You should remember this class. Okay, like for example, class, can you see this diagram? This will just give you an idea how the DNA is being replicated. Okay. Now, this is the DNA strand. Okay? This is the DNA strand. Okay, this is the DNA strand. And this strand, the double-stranded DNA is the one that is being replicated. Okay. You look at one strand when it is separated. Again, you call that the lagging strand. And the other strand, when they are separated, is known as the leading strand. Okay. And what are the enzymes involved in the replication of the DNA? Okay. One, you look at this you look at this one. Can you see this arrow class? This is the enzyme helicase. This is the enzyme helicase. And the importance, the function of this enzyme helicase, it is the one that open the DNA. The DNA double strand. It is the one that open or unwind the DNA. That is the helicase. As you can see that there is another strand, there is another enzyme here, Topo Isomerase. The Topo Isomerase is responsible for relaxing the DNA. It is responsible for relaxing the DNA in such a way that the helicase, the enzyme helicase, could open it, could unwind the DNA. So it has to be relaxed by the Topo Isomerase. And then here comes now the helicase, which will unwind the DNA, meaning the helicase will be responsible for breaking the hydrogen bond. The helicase, it is created by a helicase which break the hydrogen bonds, holding the two DNA strands together. Okay, as you can see, while the double strand is being unwound by the helicase, the synthesis of the separated strand starts. And this is brought about by the enzyme, by the enzyme known as the DNA polymerase. Brought about by the enzyme known as the DNA polymerase. Okay? So this is the DNA polymerase now. So the DNA polymerase binds and it starts synthesizing the new strand. But there is a difference between the replication of the leading strand and the replication of the lagging strand. The replication of the lagging strand, it is discontinuous. It is not continuous. Okay? Whereas the replication of the leading strand it is usually continuous. So if this is the leading strand now, in the presence of the DNA polymerase, it will keep on replicating the leading strand continuously. Continuously. There is no intermittent stoppage of the replication process. This is done continuously. Okay? The continuously. But again, class, in such a way that the DNA polymerase could replicate the leading and the lagging strands, it needs a single strands binding proteins. The single strands binding proteins. And this is responsible for making the strand linear. Linear, what do I mean by linear? Elongated. So that it could be easily replicated by the DNA polymerase. That's why it has to be straightened, the DNA, or it has to be linearized or elongated. So what will happen to the single strand binding proteins? They will bind into the lagging, into the leading and lagging strands. They will bind so that it will become, the DNA will become linear. And so that also it will make the replication of the DNA polymerase easy and straightforward. Okay? So as you can see, okay, it is just continuous. Whereas in the lagging strand, it is a discontinuous. A lagging strand, it is a discontinuous. How? First, there is the primase, the DNA primase. And this DNA primase will synthesize a short RNA primer. A short RNA primer. And then what will happen? After the primer will be synthesized, then the DNA polymerase will bind into this part. It will bind into this, and then it will start synthesizing the fragment. Okay? It will start synthesizing the fragment. And this fragment is known as the Okazaki fragment. Okazaki fragment. And then again, what will happen? Again, a primer, an RNA primer again, will be synthesized by a primase. After that, again, the DNA polymerase will bind, and then it will again synthesize a new Okazaki fragment. Okay. And this, you know, how this nick, okay? First, there is a nick in there. This nick will be joined by an enzyme known as DNA ligase. Okay? DNA ligase. Between the two Okazaki fragments. This is one Okazaki fragment, and this is another Okazaki fragment. And this will be linked together by an enzyme known as ligase. So in this case, there will be several Okazaki fragments that will be synthesized. What will happen to the Rna primer? The Rna primer will be dissolved. It will be removed. This Rna primer will be removed. Okay, and so this is now the new strand, the new replicated lagging strand, this one. And you call this the replication fork. This is called the replication fork because it is similar to a fork. Okay. So this is how the DNA is being replicated by the action of the different enzyme. Enzyme helicase, enzyme Topo Isomerase. It is, you look at class, you know the DNA it is coiled. It is tightly coiled, and the DNA should be relaxed, and that is by the action of Topo Isomerase. So this portion now of the DNA, because there is now the Topo Isomerase, it is now relaxed. And then here comes now the helicase, which will break the hydrogen bond, separating the lagging strand and the leading strand. And by the action of the DNA polymerase, a new strand will be synthesized. A new DNA strand will be synthesized. And what is the direction of the synthesis? It will be from 5' to 3'. 5' to 3'. Okay, so this is how the DNA is being replicated. Okay, now, on the previous slide, I just show you the overview, the entire view of the DNA replication. The entire view of the DNA replication. Now, steps by steps, how replication is done. Okay. Okay, as you can see in here, that there is unwinding of the DNA. It is unwind, or it is separated, forming a replication fork. And this is done by the enzyme known helicase. Okay? The first major step for DNA replication to take place is the breaking of hydrogen bonds between bases of the two anti-parallel strands. It is anti-parallel because one is 5' to 3', and the other one is 3' to 5'. They are anti-parallel. The unwinding of the two strands is the starting point. So there is unwinding. The splitting happens in the place of the chains, which are rich in 80 pair. Helicase is the enzyme that splits the two strands. The initiation point where the splitting starts is called the origin of replication. The structure that is created is known as the replication fork. Okay. So this is known as the replication fork, the origin of replication. Here, when the fork is formed, that is the start of the replication process. And that is the start on the action of the enzyme helicase by breaking down, by breaking the hydrogen bond between the two stranded DNA. Between the double stranded DNA. Now, as you can see, now, the RNA primer. Okay, how the RNA primer? The RNA primer, it is synthesized by the enzyme known Rna Primase in the initiation point of the 3', 5' parent chains. Here, the 3', 5' parent chain or strand. That is the RNA primer. The RNA nucleotide class are the RNA primer and a starter for the binding of the DNA nucleotides. So there is now the RNA primer that will bind into the lagging strand. This is the lagging strand. And then the RNA primer will be the start in the replication of this lagging strand by the binding of the DNA polymerase. We are talking here of the replication of the lagging strand. Okay? Here now. So, this is now this is the primer. You can see the primer, it is a green color. Now what happened, now this is the primer, there is now a start on the replication of this primer. As you can see, now there is a new DNA strand which is being synthesized. And that DNA strand synthesized from the RNA primer, you call that the Okazaki fragment. You call that the Okazaki fragment. So again, after the Okazaki fragment was synthesized, again, another primer, RNA primer, will bind into the lagging strand. Then the DNA polymerase will bind, and then a new DNA strand will be synthesized. Another Okazaki fragment will be synthesized. Of course, this will be open. Okay, this will be opened by the enzyme helicase. So that is what is happening. Whereas in the leading strand, there is a continuing synthesis. Continue synthesis of the leading strand. It is not fragmented. Unlike with the lagging strand, the synthesis is fragmented in the presence of a primer, RNA primer. Okay, so the DNA polymerase 1, in the lagging strand, the DNA polymerase 1 reads the fragments and removes the RNA primer. In the lagging strand, the DNA polymerase 1, remember that there are several types of DNA polymerase. The one that is used in the replication process is DNA polymerase 2. So the DNA polymerase 1 will be responsible for the removal of the RNA primer. And the gaps are closed with the action of DNA polymerase and the DNA ligase, which adds phosphate in the remaining gaps of the phosphate sugar backbone. So that is the action of DNA ligase on the gap. When it closes the gap, the DNA ligase adds phosphate in the sugar backbone. Okay. Good. This will again further strengthen your knowledge with regards to the DNA replication. The DNA replication. Okay. So this is now the leading strand. And this is the lagging strand. Okay. This is the lagging strand. As you can see, class, again, the leading strand, it is continuously replicated. Whereas the lagging strand, the replication is not continuous. Okay? There are some gaps. Okay? Because of the presence of the primer. The presence of the primer. Okay? Like for example, this one, the primer, after this fragment was synthesized, the primer was removed. Here, the primer was removed after it was synthesized. Okay, the primer was removed. And what do you call this one? You call this fragment the Okazaki fragment. The Okazaki fragment. And these two Okazaki fragments, they are linked together or joined together by the presence of ligase. Okay? This will be joined together by the presence of ligase. Okay. The same manner also with this one. Ligase, it will join by the presence of ligase. And of course, the strand that are formed, it is always complementary to the leading strand. Of course, there is the termination of the DNA replication. This process happens when the DNA polymerase reaches to an end of the strands. It is easily understood that in the last section of the lagging strand, when the Rna primer is removed, it is not possible for the DNA polymerase to seal the gap because there is no primer. So the end of the parental strand where the last primer binds isn't replicated. These ends of linear chromosomal DNA consist of non-coding DNA that contain repeat sequences and are called telomers. As a result, part of the telomere is removed in every cycle of the DNA replication. Like for example, in this case class, accordingly, this could not be sealed by a DNA polymerase, but rather this is sealed by a DNA ligase. This is sealed by the DNA ligase. Okay, so after the replication, but again, this DNA, after it has been replicated, it has to undergo repair. It has to be fixed. Why? Because there is possible errors caused during replication. And usually those nucleotides we call that wrong nucleotides. They are removed by an enzyme known as endonucleases. Those nucleotides, error nucleotides, they are removed. And these error nucleotides, when removed, the DNA polymerase fills the gaps. The DNA polymerase fills the gaps. So it is important. So there are a lot of enzymes involved in the process of replication. It does not mean after replication that the DNA nucleotides, it is now normal. There are some nucleotides that are abnormally placed during replication, abnormally synthesized during replication, and so it has to be removed. And the removal could be done by the enzyme endonuclease, meaning these are the enzymes that could cut the DNA. The enzymes that could cut the DNA. And that is called the replication. So that is the end of the replication process. That is the end of the replication process, how the DNA is being replicated. How the DNA is being copied. That is how the DNA is being copied. Okay. Do you have any question, class, about, again, DNA replication? The difference between the replication of the lagging strand and the leading strand. Now I'm going to discuss with you, again, transcription and translation of the DNA. This is just a review class, and only for the sake of our new students, I'm going to discuss again how transcription and translation of the DNA. As what I told you in my lesson in advanced biochemistry, that this is the central dogma of transcription and translation. Okay? Class, can you hear me, class? Class, can you hear
Yes.
me?
Yes, Doc.
Okay.
Yes, Doc.
Okay. The central dogma, class, always bear this in mind, that the DNA is transcribed to RNA, and then RNA is translated into protein. This is the central dogma of transcription and translation. Just remember the central dogma class in protein synthesis. When somebody asks you how protein is synthesized, you just explain it. It is synthesized through the process of transcription of the DNA into messenger RNA and translation of messenger RNA into protein. Okay, the overview of transcription and translation. The DNA, again, the deoxyribonucleic acid, it contains the information that directs protein synthesis. Cells use the two-step processes of transcription and translation to read its gene and produce the string of amino acids that make up a protein. The nucleotide sequence of a gene or a DNA determines the amino acid sequence of a protein. Okay. How does transcription differ from replication? Transcription of the DNA differ from the replication of the DNA. Again, you remember this class. It has something to do with the enzyme. Transcription is the generation of RNA from DNA, whereas replication is the synthesis of DNA from DNA, whereas transcription is the synthesis of RNA from DNA. Did you get that, class? The difference between replication and transcription. Replication, DNA from DNA. Transcription, RNA from DNA. Okay. Transcription is the generation of RNA from DNA. When we are talking of genes, we are talking of DNA. It is a process of creating messenger RNA in the nucleus from the DNA of our chromosome. Okay? From the word creating, synthesizing, making messenger RNA in the nucleus from the DNA of our chromosomes. It is catalyzed by an enzyme called Rna Polymerase, which copies the sequence of a DNA strand into a messenger RNA. Unlike with the replication process, the enzyme that is involved is the DNA polymerase. Whereas in the transcription of the DNA, the enzyme that is involved again is RNA polymerase. Okay, that is the difference. You have again to remember this class. And I keep on emphasizing to you when you took up advanced biochemistry, the difference between transcription and translation when it comes to the utilization of the enzyme. There are three types of RNA polymerase in the eukaryotes. The RNA polymerase 1, which synthesizes ribosomal RNA, and the RNA polymerase II, which synthesizes messenger RNA, and the RNA polymerase III, which synthesizes transfer RNA. So it is the RNA polymerase II that synthesizes the messenger RNA. The RNA polymerase I, ribosomal RNA, the RNA polymerase II, messenger RNA, and the RNA polymerase III, the transfer RNA. Okay, these are the three types of RNA polymerase. In the case of prokaryotic, RNA polymerase has a sigma factor. It has a sigma factor. The prokaryotic RNA polymerase, it has a sigma factor plus the core. Because without the sigma factor, the RNA polymerase could not bind into the DNA, into the promoter region of the DNA. This is the sigma factor in the prokaryotes, and this is the core enzyme. This is the RNA polymerase. Whereas in the eukaryotes, the RNA polymerase, it has no sigma factor for initiating transcription. The sigma factor class is very important for the recognition of the promoter region in the DNA. In other words, without the sigma factor, the promoter region could not be recognized by the RNA polymerase. It is also important for the melting of the DNA. And also for the recognition of terminator sequence, the sigma factor. This is in the case of the eukaryotes. There are three RNAs class. The messenger RNA again. You should also, again, remember the three RNAs. The messenger RNA, which carries the genetic information and then translate it to synthesize protein molecules. It carries the coding information to ribosomes. The genetic information is encoded in the sequence of nucleotides arranged into codons of the three bases. So that is the messenger RNA. As you can see that the messenger RNA, it contains codons, the three nucleotides, the three bases, which is called codons. Whereas the ribosomal RNA, again, it is a large RNA that make up a substantial portion of the structure of ribosomes. Central component of the ribosomes protein factory. Later on, you will learn, you will know why it is the central component of the ribosome protein factory, why it is involved in the production of protein the ribosomal RNA. While the transfer RNA is able to match genetic information and shuttle amino acid across a cell. Okay. It is the one that shuttle the amino acid or it is the one that transport the amino acid, the transfer RNA. It is the carrier of amino acid. Again, it is the one that carries the amino acid. And it contains sequence of 3 nucleotide bases at one end called the anticodon. The messenger RNA, it contains also 3 nucleotide bases, and you call that the codon. Whereas the tRNA, it also contains 3 nucleotide bases, and you call that the anticodon. And the codon and the anticodon, they have to bind together in order that an amino acid will be synthesized. Excuse me. Will be formed. In order that an amino acid will be formed or it will be synthesized. Okay, this is now the DNA. Again, similar to the replication of the DNA, there is localized unwinding of the chromosome to allow the RNA polymerase to access the DNA. Okay. In this case, it is only unwinding, but there is no, it is unwinding of the DNA. There is no complete separation of the DNA, unlike with the replication of the DNA. In transcription, there is just unwinding, and the enzyme responsible again for the unwinding of the DNA prior to transcription, it is also known as Topo Isomerase. Topo Isomerase. Okay? It's known as Topo Isomerase. And this, okay, as you can see, that this is now the unwind, just unwind prior to transcription process, but the DNA is not separated. Either strand of the DNA may be used as the template strand. Whereas in the replication of the DNA, both the DNA strands, the leading strands, and the lagging strands, those are the template strands for the replication process. But in the case of transcription, both strands could be either used as a template. Both strands of the DNA could be used as a template strand for the transcription process. What is the steps in the transcription process again? Initiation, the first step is called the initiation process. Takes place at the promoter region, the key feature for control of gene expression. And there is the promoter region again in the DNA which serve as the binding site of the RNA polymerase. And then the prokaryotes, the promoter region, you call that the box in the prokaryotes. The promoter region, you call that the box. Whereas in the eukaryote, you call that the Tata box or the CAT box or the GC box, the promoter region. Remember that this is the binding site of the RNA polymerase. Why do you have to understand very well the transcription process? The transcription process and the replication process. Because you know, like for example, like for instance, in some bacteria, when the DNA is transcribed, the bacteria DNA is being transcribed to messenger RNA. At least you have an idea how it is being transcribed into messenger RNA. And that is by the action of RNA polymerase. And the coding sequence contains coding information. And the terminator specifies the end of the transcription process. Specify the end of the transcription process, or it also specifies the end of the translation process, the mRNA transcript. So these are the important things that we should know with regards to the transcription of the DNA to messenger RNA. In the transcription site, as you can see, there is the start site and there is the terminator sequence. And there is the untranslated region or the non-coding region, and there is the coding region. And there is the promoter region. This is the gene. Supposedly, this gene setup, the setup of the gene. It has transcription start site. It has the promoter control region. It has the untranslated region. These are the DNA that is non-coded. And it has the protein coding region, and it has again the untranslated region or the non-coding, and then it has the terminator sequence. That is the end of the transcription process. And the direction is from 5' to 3' of the transcription process. The RNA polymerase uses one of the DNA strands to make RNA copy of that gene. Okay, so this is what happened. How the messenger RNA is being synthesized by the process of transcription. The RNA polymerase will bind into the DNA, and then it will start transcribing the template strand into messenger RNA by moving forward on a 5' to 3' direction. In this case, this is the template strand, and this is the one that being transcribed into messenger RNA. And this is now the messenger RNA, the transcript. Okay, so it is moving from the transcription process, is moving from 3' to 3' to 5' direction. Three prime to 5' direction. The RNA is synthesized from 5' to 3' direction. 5' to 3'. Okay. Transcription start point AUG. Gemella,
Yes, Doc.
can you still recognize this AUG? What is this amino acid? Can you still recognize that? What is that? What is this codon? What is the meaning? What amino acid is synthesized from this codon? Can you still remember?
Methionine, sir.
Methionine. Okay, so that is the start point of the transcription process. Methionine. So there is now elongation. Elongation here, as you can see, as the RNA polymerase is moving forward, an RNA transcript is being synthesized. It's being synthesized. So this is the DNA. This is the DNA, and it has the promoter region, and it has the terminator region. As what I explained, that the promoter region, this is the region by which the RNA polymerase binds the promoter. Then it will be transcribed into pre-messenger RNA. And this is the structure of the pre-messenger RNA. It has a cup, a 5-methyl 7-guanosine cup. It has an exon, it has an intron. Can you? Ever Joyce, are you present? Ever Joyce? Ever? Are you present? Absent? Peter Samonte. Peter, are you present? Yes, are you present, Peter? Can you still remember what is this exon? What is this intron? What is the difference between exon and intron? Can you still remember that? Is it the one removed in the RNA? Maria Salome. Are you present, Maria Salome? Are you present? How about Jem Taladay? Are you
Yes,
present,
Doc.
Jem?
Present po.
Maria Salome, are you present? Maria Salome. Jem.
Yes, Doc.
Joshua, can you still recall what is the function of exon and intron?
Exons are the one coding for the part of the gene that codes for proteins.
Whereas intron is what?
Introns are spliced from the
What?
are spliced or removed because it does not
Yes.
code for
Why is it spliced or removed?
because it does not code for protein po.
Okay, because that is a non-coding portion of the non-coding. It does not code for the synthesis of protein. So it has to be excised. Exon, it is the coding region of the messenger RNA. Michaela? Michaela,
Yes, Doc.
can you still remember the function or importance of this cup? Of the 5-methyl guanosine cup and the polyatyl?
It serves po as protection against the exonucleases degradation, Doc.
Oh, okay. It serves as a protection against degradation of the enzyme in the nucleases. The same, what else? Can you still recall? What else besides that? Protection from endoenzyme, endo and exoenzyme degradation. What else is the function of this polyatyl and this cup?
The polyatyl also can stabilize the messenger RNA, Doc.
Okay, it is used for stabilization. And now, on this, the mRNA now, the protein coding sequence is the one that is left behind when the non-coding intron is removed. The non-coding intron is removed. So this is now the process mRNA, and this is the one that is being translated into protein. Remember this cup class, because this cup and this polyatyl that protects the messenger RNA. It is a protection for the messenger RNA from degradation of this endonuclease enzyme. Okay, so this is the processing of the transcribed messenger RNA. This is now the mature mRNA, and this could be transported into the cytoplasm. This could be transported or exported to the cytoplasm once it is processed, where translation will occur. Okay, this is just a review, the importance of the cup, 5-methyl guanosine cup. Okay, so it is used for recognition as well as protection from exonucleases degradation. Whereas the polyethyl, it stabilizes the messenger RNA, and it also prevents ribonuclease degradation. I will not anymore deals with this how introns are removed. Then the termination class of the transcription process when it reaches the termination sequence. When it reaches the termination sequence. So the transcript or the messenger RNA will be released from the DNA template. And this is the difference between the transcription process of the prokaryotes and the eukaryotes. In the eukaryotes, the transcription and the translation process occur in the cytoplasm. But whereas in the eukaryotes, in the eukaryotic cell, the transcription process occur inside the nucleus in the eukaryotic cell, and the translation process occur in the cytoplasm. This is the difference between the transcription and the occurrence of the transcription and the translation process of the DNA and the messenger RNA between the prokaryotic cell, the bacterial cell, and the eukaryotic cell. So this is what happened. The DNA is transcribed into messenger RNA, and then the messenger RNA is being processed. Then it is transported or exported into the cytoplasm, where it is in the cytoplasm again, where it is translated into protein. This is now the polypeptide, where it is translated into protein by the action of the three RNA, the ribosomal RNA, the messenger RNA, and these are the transfer RNA. The transfer RNA, and these are the new proteins that have been synthesized, the polypeptide. And this involves processing. it has to be processed, further processed, and those proteins, those amino acids that are not necessary has to be degraded or it has to be removed. In other words, this polypeptide will undergo modification. It will be modified. Okay, and that is transcription class. And that is transcription. I think I have to end up here, and then next meeting I'm going to continue my lecture. I'm going to continue my lecture next meeting class about translation, about again, about how, the DNA